Cancer is a hard problem, in part because cancer is not just one disease, but a class of over 100 diseases that all manifest as uncontrolled cell growth. Normally, cells grow and divide at a rate that maintains a balance in the body. However, in cancer, this balance is disrupted, leading to disease. There are many factors associated with cancer, some that we can control, like smoking, and others that we can't, like family history. But the underlying cause is often a genetic change which led to disease. The cell is a complicated place, but it's highly organized, and there are groups of genes responsible for different processes in the cell. Some enhance and others inhibit each other's function to maintain a correct balance. But when one of these genes misbehaves, it can have many effects on other genes that it talks to, possibly disrupting the function of the overall process. Likewise, all the different processes in a cell also talk to each other. So when one process is disrupted, it may affect other related processes as well. In order to understand cancer, our goal is to understand the specific changes in the cell that give rise to disease. Using a current technology called microarrays, we can measure the activity of many genes at once. Microarrays contain light-emitting dots for each gene that light up brighter for genes that are active and dimmer when they're not. This gives us a way to assign a number to the activity of each gene, which allows us to use computer algorithms and statistics to look for meaningful differences between cells and between conditions. For instance, we can compare cancer cells with non-cancer cells. Or, in our project, we are looking at cancer cells that are treated with a drug versus cancer cells that are not. A microarray measurement is a snapshot of gene activity taken at a specific moment in time. Looking at one microarray is like looking at one frame of a movie and trying to understand the whole plot. Like, take this guy. Is he really levitating a potato? When viewed in context, never mind. In order to see what's really going on, one has to look at changes over time. In the same way, looking at multiple microarrays over time gives us a better picture of what is really going on. In this project, we used a dataset that was generated to better understand how a new cancer drug works. In the experiment, cancer cells in the laboratory were treated with a drug and had microarray measurements taken at a series of time points after treatment. Similar measurements were taken for untreated cells. Our task is to identify differences in gene activity between treated and untreated cells. To make sense of this data, we considered all genes at all time points and identified important genes by whether they are significantly different between conditions and across time points. We then grouped those important genes into categories based on their function, and finally visualized the data to make sense of patterns we observed. This way of summarizing large amounts of biological data is necessary, but visualizing the information to easily understand it is a major challenge. Our tool implements a pipeline that takes time-course microarray data, analyzes it in the way we described, and visualizes it in an interactive web page. It is designed to explore all the data at different levels of detail. The software is currently under active development, and we expect it to be a valuable tool in exploring our dataset and other time-course datasets.